Tēnā koutou katoa. Thank you so much for allowing me the privilege of being here with you today to speak to you. Apologies for not being here in person and for being on a virtual platform, but thank you so much for allowing me to share my story. My name is Suzanne Rowe, and I'm talking on behalf of a large team of scientists who've been working very hard to apply genomic technology and engineering to help solve the problem of methane emissions or greenhouse gas from ruminant livestock. Ruminant livestock is a big issue in terms of the greenhouse gas profile of New Zealand. Half of our greenhouse gas emissions come from agriculture, and 73% of those come from enteric methane from livestock. And what do I mean by enteric methane from livestock? Well, when a ruminant takes in complex starches, so plant cell walls, she can't break them down by herself. So she has a symbiotic relationship with microbes that live in her gut. Those microbes in her gut, they break down the grass, they break down the cell walls, and they ferment her feed for her. So they get a great home. She in turn gets her feed fermented and an energy source. So that fermentation creates volatile fatty acids, a carb source, if you will. And these volatile fatty acids are then provided to the animal to grow, to lay down meat, to produce milk. But whilst this fermentation is in play, gas is given off. And one of those gases, hydrogen gas, accumulates in the rumen, and it needs to go somewhere. So over 40 millennia, ruminants have evolved this great system where the hydrogen is combined with carbon and it's expelled from the rumen. So it's belched out, as it were, from the rumen, comes out of the front end as a CH4 or methane. Now, obviously, one of the easiest ways to produce less methane from ruminants would be to have less animals, to have less animals on the planet. But of course, life is never that simple. We have so much pasture land across the world, and the Food and Agriculture Organization recognizes very, very clearly that within their sustainable development goals, that action on climate change is needed. It's urgent in livestock, but it must not come at the expense of ending hunger. And livestock, they believe, can really help to achieve a balance where we feed the hungry and the malnourished, but we minimize greenhouse gases. And that's where we come in. Our quest was to find whether individual animals produce less methane per mouthful of feed eaten. So are there differences? There are differences in every niche of life. And are there differences in methane? And if so, can we capture them? Can we harness them? And essentially, can we breed for them? So what was done 15 years ago, started by a man called John McEwen, was that sheep across New Zealand were gathered together, and 1,000 were chosen. 1,000 were chosen that were representative of the national flock, and they were screened through respiration chambers. It was a very expensive, very time-consuming, very arduous process. It took around 4 or 5 years. But what he really successfully managed to do was screen off the highs and the lows. And over 3 generations, the highs have been mated to the highs and the lows have been bred to the lows. And what we have found is that every single year, the offspring of these animals get further and further apart. So the lows mated to the lows give us much lower offspring. And it's been going on over 3 generations. And now the average of the flock is 12% different. But if I took my highest emitting ram and my lowest emitting ram where the selection pressure is greatest, I see a 30% difference. So real evidence, real on-the-ground evidence that it's a genetic trait, that the animal is in control, and then it's passed on to the next generation, and that we can breed for it. So the next objective for us was, what are we doing? We're creating these divergences in these flocks. What's happening inside the sheep? So we looked very carefully at all aspects of the sheep, and what we saw was the low methane animal, she eats less. She eats less than her high emitting counterpart at each meal, but she eats more meals. So actually she eats more across the day. So she's having these small meals, but she's having frequent meals throughout her day. She actually has a smaller rumen, but she has more advanced papillae. They're denser and they're longer. She has more wool. So obviously there's something going on in the gut that's providing the nutrients that she needs to grow more wool than her high emitting counterpart. And that's the crux of the matter. The crux of the matter is that her microbial fermentation, the microbes that sit in the low animal's gut, are different. She's housing a different microbial community, and she's controlling that herself, which is really quite amazing. And what that microbial community is doing is changing the ratio of the energy source to the animal. So it's changing the fermentation, changing the amount of what we call propionate that's given to the animal. And that's doing is consuming hydrogen. We're getting less hydrogen, so we're getting less methane. And at the same time, we're getting less acetate and butyrate. So what we're actually getting is less fat on the animal. So we get a leaner, more muscular animal. She's actually very profitable. She's more profitable than her high-emitting counterpart, but she is quite different. But we run these animals in the field. We run them all together. The highs and the lows are run as a single flock. The only time we separate them is at mating, and you cannot tell the difference on the ground. So you couldn't walk amongst this flock and find those low emitters. You simply have to measure them. And how do we measure them? How do we go along with measuring and choosing the lowest and choosing parents for the next generation? Well, what we did was we went back to those respiration chamber experiments, which are actually done at 6-minute time points, and we found the best time to measure. We found the 1 hour in the day across all those 6-minute time points that gave us the most information and was most correlated to 24 hours per day. And we took that 1 hour, and then we had our engineers design us a portable accumulation chamber that will capture the gas given off a sheep in a single hour in a very, very simple way. So this is a portable accumulation chamber. It's a very simple box. But what we do is we calculate the volume of the animal. We measure her for an hour using a piece of technology that's come from the mines. So this is a very, very simple piece of technology, but a very sensitive piece of technology that will detect for us when we draw a sample out of this box. It draws methane, carbon dioxide, and oxygen. And we can use the temperature around the animal, we can use the pressure on the day, we can use her volume, the volume of air in the chamber, and we can estimate in parts per million the methane that she's giving off, and we can convert that to grams per day. And now we know how she ranks with her contemporaries. So we can actually find the lows and we can find the highs on farm in a simple 1 hour measure, and we only need to do it once in her entire lifetime. So that's really quite an incredible advance. We've gone from understanding the genomics and the physiology to engineering a solution that we can use on farm. And we wanted to go much further than that. We wanted to use it not just on a research farm, but across New Zealand. So our engineers, again, mounted these on a trailer. You may have seen this coming up and down the road. If you do, give us a wave. So what we've done is we have this trailer. It has 12 units on it, and we can go anywhere across the country and are doing so. And we're measuring sheep for farmers. We're getting out on farm and we're measuring them. And we've teamed up with Beef and Lamb New Zealand Genetics, and they supply breeding values using genomic profiles for the other traits in the sheep industry. And what we've done is we've aligned ourselves with them so that we deliver those methane measures from farm into their system. And when breeders get a breeding value or they get a list of how their animals rank, their genetic merit, their breeding worth, they get methane too. So not only now can they choose animals for their productivity, for their resistance to disease, or whatever trait they feel is important within their system, they also get methane. So they can choose to select for low methane should it make sense to them. And that's really exciting for us. That's a big development. And I just want to talk a little bit about what that implementation looked like, because these bars are flocks. So each of these colored bars are a flock. And in 2018, we went mobile. We engineered a trailer and we got on the road. So that meant that the next year there were a lot more flocks. But then we got industry funding, we got industry support. So breeders didn't have to pay for quite so many measures. So at the time we were measuring sheep twice. We were measuring them, then we were going back 2 weeks later, measuring them again. That was hard. It was tough for the farmer to feed them really, really well for that entire period. So we got a consistent measure. So what we did was go back, wrap some science around it, evaluate the data, and say, hey, if we measure a few more animals, we can, around 15% more animals, we can drop the second measure. So we went from engineering the technology, having economic support, to scientifically evaluating what was going on, dropping a measure, and now we're up to as many as we can measure. So we're currently up to 5,000 animals a year, and our books are full. So we'll be engineering another trailer, and we're off. And the green bars at the bottom are just to show you the range. So the range goes for, it's around 30% of the average. So it goes from about 2 grams less than average for an animal, up to around 1.5 grams higher than average for an animal. And we see that spread in every flock we look at. So every flock that we look at, every flock that we go into has low emitters in it that can be chosen. And that's really exciting. That means that farmers have it in their hands to be able to do something for themselves. And it's not just our farmers. When we talk to our peers across the world and we've joined forces, we have global collaborations and their ordering chambers, we're sending them, we're helping them with technology, we're helping them with the operating practices, and together we've gone global with this. So that's really exciting. But if we come back to a New Zealand context, if we think about what we're actually achieving here, we're actually saving, we think by 2040, around 4.5 million tonnes of CO2 by implementing this across breeders' flocks and saving around 0.5% per year in the national flock. So a really, really modest change. And why so modest? Because that doesn't affect production. That means that we can not impact hugely production. We don't take selection pressure off production. So what we can do is implement this, and excitingly, it doesn't cost very much. It costs about $1.72 a tonne of CO2 equivalent mitigated, and the returns are really good. So it's a very low cost, very simple, very straightforward, very easily marketed way of reducing methane at the farm gate. So that's sheep. We're really excited about the story in sheep, but what about cattle? 68% of New Zealand's emissions from enteric emissions from livestock are from cattle. 62% of the world's are from cattle. So we need to do something in cattle. What can we do? How can we transfer this technology, all these developments in sheep? Well, we have a Global Research Alliance livestock research flagship. And what we've done is we've taken what we know about the microbes, those microbial communities. Now, we take a sample from the rumen using a simple stomach tube and we sequence it, and we can use the information that we get from the DNA profiles from those microbes to build a picture about the animal. And what we can then do is predict how much methane she's going to emit. And we've been doing this with cattle across the world, and it's been really successful, and successfully showing us that we can transfer our learnings in our microbial profiling and our prediction, our statistical techniques into cattle. And here we are on farm down in the Te Anau Basin, sampling some cattle. We've developed a whole system where we have barcoded tubes. They go through the lab, and we have a whole system in place to do that. We've also developed portable accumulation chambers for cattle. This is hot off the press. We have one. I say we've developed chambers. That's not true. We've built one, and we've tested it. We put 30 dairy heifers through. They went through their second time last week. We borrowed them from a local farmer who was really excited to help us with our research, and it went spectacularly well. So I think there's going to be a place to get a direct methane measure from these chambers with cattle on farm too. So that'll be a much bigger trailer going past you. So give that one a wave too. And finally, the next generation of predictors. How can we combine everything we know about the microbes, about the methane, about the physiology, about these fatty acid profiles that I said are coming from the rumen, giving the animal energy? Well, what we know is when we really drill down, when we look very closely at the specific fatty acid profile of an animal in terms of its meat and its milk, we catch that signature. We can see it. We can see the low methane animal having a different fermentation, providing this propionate, and then having a leaner animal with a different fatty acid profile. And we can use infrared technology to catch that profile, and we can use deep learning and statistical techniques to then predict. So the holy grail for us is to be able to test an animal without touching an animal. So to test that animal with using meat, using milk, and actually be able to predict in thousands or even millions of animals. So that's where we are at the moment, and that's what we're striving for. So next steps for us in terms of cattle are international collaboration, understanding that physiology and how it works across a whole range of cattle and species. Delving deeper into the genomics, the methane, the rumen microbes, and finally that milk profile, and seeing if we can come up with a universal predictor that can be used as a high throughput and low cost predictor, and is accessible to any country in the world, and really understanding that microbial prediction. But really what we want to do is also understand additivity of strategies. We know that breeding is not the only mitigation approach. In fact, it's the most modest. We have been tasked with a 50% reduction by 2050. It's unlikely we'll get there by breeding alone. There are feed additives, there are feeding strategies, there are vaccines, there are many more things not implemented yet, but in testing. What we want to know is how they combine in a system. What happens if I breed a low methane animal and then I wrap all these mitigation approaches around her? So one of the things that we'd like to do on an AgResearch farm is to have a demonstration combining these low methane animals with different feeding, different additives, the vaccine when it becomes available, and really demonstrating what happens when we combine these technologies. So we still have much to do. We're far from finished, but I will finish there because I only have a short amount of time with you. And just to acknowledge the financial support, which has been immense, and as I say, has been going on for 15 years. The Pastoral Greenhouse Gas Research Consortium, a group of industry partners who have come together to help implement the New Zealand Agricultural Greenhouse Gas Research Centre, the Ministry for Primary Industries, Beef and Lamb New Zealand, the Global Research Alliance, and all these funders who have worked so hard alongside us. But finally, and most importantly, those breeders who took a chance, who were out there on the edge and said, yep, we're with you, and we'd like to try it. So a big thank you to those guys, and thank you so much for listening.